Yes, sir.
Okay, nervous tissue. Let's start the nervous tissue topic. Nervous tissue. So you know what is a nervous tissue. I guess you understand. Is a transmit. So is the main function of nervous tissue is a employ modifications in membrane electrical potentials to relay signals throughout the body. So this when signals goes very fast way, so in a very rapid way. So from one side to another side, it is made by nervous tissue. So neurons, and usually we talk. You heard about neurons, cells of neurons, and we will discuss neurons. But also we have some additional tissue in nervous tissue that we will discuss very soon. Okay, neurons. They relay sensory information from the internal and external environments. So it's like sensitive neurons we have. We have integrate information among millions of neurons. It's integrative neurons, so which switch signal from gained signal from one side to another. For example, in brain we predominantly possess integrative neurons. And third one is the motor neurons. They transmit effector signals to muscles and glands. They are motor neurons, which facilitate the normal secretion and contraction. So anatomical subdivisions of nervous tissue. We have a central nervous tissue and peripheral nervous systems. You have central nervous system and peripheral nervous system. Central nervous system is composed of brain and spinal cord. So you know it. So this is here we have a brain, and this is spinal cord. This is central nervous system because here we have integrative neurons predominantly, and peripheral nervous system, which composed of nerves and ganglia, which are predominantly, so not predominantly, always composed of effector neurons or effector or affective neurons, like sensory neurons. This is peripheral nervous system. So peripheral nervous system is composed of nerves and ganglia. Ganglia, you will discuss it in anatomy, I guess, but they're present. For example, we have a dorsal root ganglia, sensitive for sensitive neurons, and also we have sympathetic and parasympathetic ganglia, which appear in different locations. Usually, sympathetic nervous ganglia they're present near the central nervous system. Parasympathetic ganglions are present in intramurally two organs. That's the main idea. Physiological role of them you discuss in physiology. Cells of nervous tissue. We have neurons and supportive cells. So neurons, functionally, we all discussed it. They are functional units of nervous system, and all the effects we heard about nervous tissues, the transmission of signals and information to one neuron to another, and also to muscles or glands, signals we directly shared. They are made by neurons, and they facilitate this joint one neuron to another with the help of synapses. With the synapses, and neurons possess dendrites and axons. Dendrites and axons, which are attached to cell body, and they facilitate the normal joining of one neuron to another neuron. It's a stable containing. So how it's arranged, we'll discuss. We'll discuss it right now. And then they form complex and highly integrated circuits. All the circuits, all these reflect arches you discussed in physiology, um, they are made up of neurons and axons and dendrites of these neurons. Okay, let's join some students. Okay, supportive cells. But we have a lot of supportive cells because neurons are very gentle. Neurons are it's like noble persons. Yeah, you know it's. I usually compare it. Indian students understand it well, but you also understand the Indian system. Yeah, so Brahmans, neurons are like Brahmans. They are the main and most important and very how it's called, very gentle persons. And they provide the most important function, so like spiritual function of the society. Yeah, but to facilitate the function of neurons, we need to possess supportive cells which help them, and without supportive cells these neurons cannot act. The supportive cells, they serve to the neurons, and only in this case our system is working. For example, okay, let's talk about this. Supportive cells are outnumbered. They are 10 times more supportive cells than neurons, and usually students forgot about the supportive cells, but they play a very important role, and especially in diseases of nervous system, supportive cells play most important function. Okay, supportive cells also provide metabolic and structural support for neurons, and they provide insulation. Also, they help in homeostasis and phagocytic function of the nervous tissue. So, yeah, for example, myelin sheaths, you learned, yeah, in nerves we have myelinated fibers and non-myelinated fibers, and they're made by supportive cells. Okay, for example, Schwann cell, which you discussed in physiology, I guess, they are supportive cells. Okay, and then comprised of astrocytes, oligodendrocytes, microglia, ependymal cells in CNS, and Schwann cells in PNS. So so you see, you have to remember these cells, supportive cells in the central nervous system: astrocytes, oligodendrocytes, microglia, and ependymal cells. Okay, let's talk about them. Let's talk about typical neuron structure. You discussed it in biology, discussed in physiology, and here I must just remind you what is a typical neuron. Here you see cell body. This is cell body, which is composed of nucleus and cytoplasm. So this cytoplasm, and in cytoplasm you see it possesses well-developed cytoskeleton. Well-developed cytoskeleton, it possesses specific intermediate filaments which are known as neurofilaments. These neurofilaments are very important in detection of tumors. So some tumors of nervous system, they have specific neurofilaments, and we use specific stains to detect these neurofilaments, and we can consider, yeah, this tumor grows from neurons. So very rarely, but we have these types of tumors. Okay, and also in cytoplasm we have microtubules. Microtubules, which facilitate the activity, so which helps in switching of one neuron to another neuron, and this growth of these microtubules responsible for attachment of them. So yeah, and one thing I forgot to say about smooth muscle and to say about skeletal and cardiac muscles is, okay, neurons, skeletal muscles, and cardiac muscles are unable for cell division, almost unable for cell division, almost. So yeah, very rare cases, so few mitosis are present in neurons and in cardiac muscle and skeletal muscles. So smooth muscles is exception here, because smooth muscles are able for cell division very slowly, but they can divide mitotically. But neurons are almost never divided mitotically, and death of neurons is irreplaceable. Death of neurons. So when one neuron died, so some function of the brain is interrupted. So that's main idea. But we can damage the axon. Okay, let's talk about this. Um, let's talk about this later. Okay, in cytoplasm we also have a rough endoplasmic reticulum, and polysomes. Polysomes is a polyribosomes. There are ribosomes which are joined in a large structure. Yes, it's like granules. And these granules, these polysomes, forms a specific structure which is known as Neisel substance. Neisel substance is present. Is present all over the cytoplasm. But is not present in axon hillock. So let's talk about later. It possesses well-developed Golgi apparatus. A well-developed Golgi apparatus is responsible for transmission of signals, transmission of material from one side of cell to another side of cell, and to release some compounds outside. So it is responsible for formation of cell membranes and cytoplasm membranes and other innate membranes like smooth endoplasmic reticulum and rough endoplasmic reticulum. So this is a Golgi apparatus function, and it also takes part in formation of some secretory compounds, so like mediators of nerve signals. Okay, and they possess numerous mitochondria. So neurons possess a lot of mitochondria, because main function of neuron is a sharing of active potential. But active potential is a passive process. Yeah, but depolarization is passive process. But repolarization is a very active process. It requires very large amount of ATP to facilitate the sodium-potassium pump activity. And sodium potassium pumps the potassium back and leave the sodium outside the cell, and which change back the normal state of resting potential. So that's a role of this mitochondria, which are very abundantly present in the neuron. Yeah, and that's why actually, so one more thing, that's why 20% of glucose of the body, 20% of glucose is used by our brain. And that's why you might heard that thing that before the exam, before the model, you have to eat something sweet. Yeah, you have to eat something with a lot of glucose inside, and only and with a large amount of glucose, we can handle the normal action of the brain. And also might be heard about the stirring so it's called um, for, so in stirring conditions, so we lose our conscience. Yeah, so it's losing of consciousness due to stirring is very common, especially in I heard about it. One my teacher said to me that one clinical subject teacher, he said to me that we have in his classes, in his lectures, sometimes a lot of people they fall down because of starving. Yes, because they didn't take breakfast, because they need to use their brain. Yes, they need to think about it. They have a very large load to the brain, but glucose storages are empty, and that's why his students so falling down and with the um, they get unconsciousness, so because of glucose deprivation. Okay, let's talk about outgrowth of the neurons. They are dendrites. Dendrites, it's derived from word dendros. It's tree like. So because it possesses a lot of branches, and this dendrite's main role is gain signal from somewhere else. So it gains a signal from surrounding tissue. So, for example, another neuron, another axon might interact with it, and they cause the irritation of these dendrites, for example. And then cell body decides, yeah, so should we transmit the signal or not? So, okay, we'll discuss it now. So in dendrites, organelles of dendrites are similar to cytoplasm. There are microtubules and neurofilaments, rough endoplasmic reticulum and polysomes are present, smooth endoplasmic reticulum present, and mitochondria present there. So you see, same arrangement as cytoplasm. So we can say that dendrites is just outgrowth of cytoplasm. That's it. Okay, but what? Let's talk about another structure, which is axon. Axon is a Okay, take a look. Axon is usually one per one cell. Usually, so I said we'll discuss some exceptions, but usually one neuron possesses only one axon in most cases. And axon initiated from axon hillock. Axon hillock, it's outgrowth of cytoplasm, which is axon hillock. It's a region of cell body where axon originates. It's devoid of rough endoplasmic reticulum, so it's absent of rough endoplasmic reticulum, and you see no nasal substance in axon hillock, and continues with initial segment of axon that is highly electrically excitable zone for initiation of nervous impulse. So take a look. Okay, here. You see, and now you see the features. Yeah, so you see dendrites are activated, and then signals collecting in the cell body, and actually they are collecting in axon hillock. And if this irritation which made by dendrites, they're able to irritate axon hillock, they're able to stimulate the axon hillock to transmit the signal. Axon hillock continues the signaling along the axon. If this amount of irritation is not enough to initiate the axon hillock transmission, this axon hillock just silent, and axon is not irritated. That's how the thoughts are made. That's how our stimuli made. So it's just I give you example. Yeah, for example, if your subject of study is very easy, yeah, So it's very easy, and you know that, yeah, I know, for example, I know, don't know, biology. Yeah, from school you learn biology very well. Yeah, you don't need preparation for the subject. Yeah, you understand that some points you skipped. Yeah, in biology, but your basic knowledge is enough to submit the model. And this irritation of model is not enough to stimulate you to learn something. It's not enough to open the book. Yeah. But if it's, for example, some new subject, for example, okay, embryology. Yeah, you never had embryology before. You don't know embryology totally, and you must open the book, yeah, before the model. And this irritation is very high, and then it gives you the stimuli to start the preparation. Same in exon hillock activity. So if irritation is enough, here we have something like switcher. So you can remember exon hillock is like switcher of exon. So if irritation is enough, exon hillock irritates, and exon conducts the signal to effector cell. If it's not enough, so it remains silent. That's why the stimuli is not enough to transport. And this is very good for us because we have a cacophony of signals in our brain. Not every of the signal is enough to provide some activity. For example, right now, some of you want to eat something. Yeah, some of you want to skip the class, but you understand. So if you skip it right now, so it will be recorded in a lecture, and everyone can see that you skip the class. And actually, in the end, in the semester ending, so I can mention it, and then I will be more strict to you on exam. So that's you understand this opinion, yeah? Okay, then exon. Exon by structure is similar to exon hillock. Exon also possesses no rough endoplasmic reticulum. It possesses only smooth endoplasmic reticulum, cell membrane, a lot of mitochondria. It possesses microtubules, also possesses a lot of neurofilaments. And in the end of exon, we have synaptic terminals, synaptic synaptic knobs. They're known as synaptic knobs or synaptic terminals, synaptic endings. It's a similar name, different names of similar structure. It's a synaptic bottom. Sometimes you can hear this, which are present on effector cell. It might be it might be dendrite of another neuron. It might be muscular cell. It might be glandular cells. Which stimulate the secretion. Okay, about synapses we'll discuss a little later. Okay, neurons. There are three main types of neurons. Most common type, and which is present almost everywhere in the central nervous system, and in a large variety present in peripheral nervous system, it's a multipolar neuron. Multipolar neuron possesses a lot of dendrites and one axon. This is typical appearance. And in the end in the end of axon, we have a synaptic vesicle, synaptic node. Okay, usually they're afferent neurons or integrated neurons. They're present in central nervous system and in motor neurons. Okay. Then we have pseudo-unipolar neuron. Pseudo-unipolar neuron is exclusively present in sensitive neurons. So you see, it is a pseudo-unipolar neuron it is a exception. So usually we used to think, yeah, we have one axon and it has one direction. But in pseudo-unipolar neuron, so here we have the axon hillock. This is axon hillock on this location. But then axon hillock is continued into two axons in one direction and another direction. And actually passaging of signals goes along with it without interruption. So all the sensory information we have through the pseudo-unipolar neuron reach the central nervous system. Pseudo-unipolar neurons are present in sensory neurons. All the senses we have from your skin, or all senses of touch receptors, we will discuss in skin topic. They are made by pseudo-unipolar neurons. And for example, this ending is a sensory part, and then transmission goes through the central part, and it afferent part so reach the central nervous system. And bodies bodies are present in dorsal root ganglia. Might be heard about dorsal root ganglia in physiology. So dorsal root ganglia is a typically cell bodies of pseudounipolar neurons. But axons of the pseudounipolar neurons are sensory axons of the nerves. So this half of nerves are made up of this pseudounipolar neurons pseudounipolar neurons axon. And bipolar neuron. Bipolar neuron is the most ancient type of neuron. So actually, most of our neurons of the body, this initial stage of development of them. So it possesses one cell body, one dendrite, and one axon. Yeah, but during the development, we can produce more dendrites, much more dendrites, but axon still remains just one neuron, just one axon is remained. But it is found in some specialized areas, like in organs of special sense. The retina of the eye, olfactory epithelium, vestibular and cochlear ganglia of the inner ear. So the special senses, all of them, they are bipolar neurons. They have only one receptor and only one axon. Okay, arrangement of neuronal cell bodies and their processes. So it's in central nervous system. This group of cell bodies, they're located in nucleus or cortex. That's arrangement. In peripheral nervous system, the cell bodies are present in ganglions. Okay, but what about bundle of processes of the cells? In central nervous system, these tracts, they form the tracts inside the central nervous system, and these tracts of central nervous system, they are named as white matter. Yeah, white matter. In so, for example, groups of cell bodies, they're gray matter. Yeah, and you see, gray matter is a thinking part, white matter is a part of transmission of signal. But in peripheral nervous system, once again, groups of cells are known as ganglion. So like dorsal root ganglion we discussed right now. And bundle of processes are known as nerve. Let's talk about the synapse. So what is a synapse, or in some spelling, some pronunciation is synapse. So both of them are right. I checked it out. And what is the point? So you remember that synapse is the ending of all axons. And this is axon terminal. And when active potential reach this part, so what happens inside? Actually, on synaptic membrane, so in the end of axon terminal, we have a calcium channels. And when depolarization wave comes to these calcium channels, they get opened, and calcium, which is located extracellularly, enters inside. And calcium inside the cell, so in biochemistry you will discuss it, they initiate the process of calmodulin activation, and due to this, so vesicles which are collected in cytoplasm, they are transported towards the cell membrane and release it outside. It is might be, if you remember the previous semester, it's a regulatory pathway of excretion, regulatory pathway of exocytosis. So when calcium influx followed with the releasing of vesicles, and due to this, and what do these vesicles contain? Vesicles of synapses, they contain the neurotransmitters. And most common type of neurotransmitters is acetylcholine, and might be noradrenaline, and we have a numerous amount of this neurotransmitters, but most commonly it's acetylcholine. And due to this, acetylcholine releases into the synaptic cleft, and acetylcholine attached to receptor of the effector cell. It might be another neuron, it might be muscle, it might be a gland cell. So, but anyway, it releases and promotes the activity of adjacent cell. Okay, in Google, in Wikipedia, you can find that we have different types of synapses. We have chemical synapses and electrical synapses. But in human body, we have only one type of synapses. It's a chemical synapses. Once again, only chemical synapses. So that's why if in exam I heard about this, that we have electrical synapses, so I put 2 mark, because we don't have electrical synapses in human body. We care about only humans here. It's not biology, it's human histology. Yeah, so it's only chemical synapses are present here, and receptors is here on the surface of postsynaptic membrane. We have receptors, and they're activized. Again, what we have? So axon terminal. So here presynaptic membrane. Presynaptic membrane. On presynaptic membrane, we have calcium, voltage-gated calcium channel. Due to depolarization wave, calcium channels open, calcium influx inside the axon. Then this calcium causes the activation of regulatory pathway of exocytosis, and the synaptic vesicles exocytosed and released outside their secretion directly in the synaptic cleft. This one is known as gel-like component, is known as synaptic cleft between the presynaptic membrane and postsynaptic membrane. And then it activates the receptors, which are acetylcholine receptors or another types of receptors on the surface of postsynaptic membrane. Okay, once again, presynaptic membrane, synaptic cleft, postsynaptic membrane. Before the presynaptic membrane, we have synaptic vesicles due to depolarization. Vesicles released in the synaptic cleft, and synaptic cleft is semi-liquid, and these compounds easily reach the receptors of postsynaptic membrane. Okay, I hope now you got it. So synapses are classified according to postsynaptic target. It might be axodendritic, most common type, so most classical type. Yeah, so one axon and so attached to dendrite, and this transmission goes from one neuron to another neuron. We have axosomatic. So this is a specific type of neurons, synapses which are attached, which are directed to the body of neuron. So because the dendrites, you see, it's additional structures. The dendrites are they cannot directly stimulate entire cell. It gives just some part of stimulation. But axosomatic dendrites, here axosomatic here is present. Axosomatic synapse, or this one is also axosomatic. It directly stimulates the cell or shuts it down. So actually, we have this feature too. So to depress the activity of this neuron. So also happen with axosomatic synapse. I also have axonic. Axonic synapse is well shown here on this diagram. You see, axonic so happens so also to depress the depress the synaptic transmission. So before it causes repolarization. It causes a repolarization before the activation of synaptic knob, and synaptic vesicles are not released outside. This is axonic synapse, usually present here, usually present for this purpose. And also one more synapse classification is neuromuscular junction. Neuromuscular junction is a usual type of neuron, usual type of synapse, but it is attached not to another neuron but to muscle, and they stimulate the skeletal muscle activity. So they are present in skeletal muscles. And also we have dendrosomatic somatosomatic synapses, but they are very uncommon. Structure of the synapses. So okay, we discussed it here. Okay, I want to explain you the one specific type of synapse known as Bouton and Passan. So it's a French name of this French name, which says that it's Bouton-like swellings along the length of an axon. So I can show it. For example, okay, just imagine that these axon terminals are present not only in the end, but they're present in the middle of axon, and along all the axon we have the synaptic vesicles, synaptic knobs. And due to this synaptic knobs, they can activate. So one neuron can activize a lot of different cells. So one neuron activize. So especially it's present. Sorry. Especially it's present Bouton and Passan, they're present in smooth muscle innervation. Smooth muscle innervation. So when we need, now we discussed, yes, a smooth muscle. They have a very restricted features of transmission, of signal transmission. That's why we need to arrange it with neurons. Okay, this diagram also contributes this presynaptic membrane, postsynaptic membrane, synaptic cleft, synaptic vesicles, so mitochondria which produce ATP, so terminal, dendrite of second neuron. So we discussed it yet. Reflex arc. In physiology, we discuss reflex arc. So, for example, we have receptors on skin, and you feel like something or pain or might be heat, yeah, and it gives a signal to dorsal root ganglia and through the sensory neuron. You remember it's pseudounipolar neuron present in sensory neurons. Pseudounipolar neuron transmits the signal to the association neuron. It's a association neuron. It's a neuron of so integrative neuron. Sometimes it's called as integrative neuron, and then is this association neuron so switch to motor neuron, and motor neuron causes the activation of skeletal muscles or smooth muscles or any other reflex, so any other activity which is made by motor neuron activation. Supportive cells. So now I'll so wait a bit. So I understand that time is very shrink. So we have 10 more minutes, and I'll try to arrange this in 10 minutes. Supportive cells. So this diagram explains all supportive cells which we have here. So you have this PPT, so you can watch it these definitions. My attention it will be concentrated on this diagram only. So first of all, it's astrocytes. Here you see astrocytes, and you see according to this diagram, astrocytes, their main function is the joining of blood vessel with a neuron. Joining of blood vessel with neuron. So, and you remember that blood vessels, they share the nourishment material. So nutritional material and oxygen is shared by them. But also along with the blood, so in the blood we have a lot of wastes. We have a lot of potentially injuries to neuron substances, and that's the function of astrocyte is to take everything and distribute only necessary for neuron compounds to neuron. Everything which is not is potentially dangerous. It will be kept by astrocyte or released outside, released back into the blood vessel. So it doesn't allow the direct connection between blood and neuron, because as I said, neurons are very noble cell. It's a one of the it's a Brahman cell, yeah. So that's it doesn't, it shouldn't be involved in troubles of blood, yeah. So that astrocyte facilitates this. They just shift the most, so share the most delicious things to neuron. If it's not delicious, it doesn't share it to neuron, because neuron can die if it doesn't get proper amount of nourishment, and proper nourishment we require. And astrocyte, so you see by shape, it is like, it also resembles a neuron, but you shouldn't confuse it. It's astrocytes are servant cell. It's a supportive cell. Okay, another cell type which is present in the central nervous system, it's oligodendrocyte. Oligodendrocyte also possess a large outgrowth of cells, and these outgrowths they just wrap around the axons, sometimes even dendrites, but actually usually axons of the neurons, and they form the myelin sheath. One oligodendrocyte may serve to a lot of a lot of axons. So so you see, once again, one oligodendrocyte serves to a lot of axons, which differs it from a Schwann cell. Actually, oligodendrocyte and Schwann cell are very close relatives, but oligodendrocyte serves to a lot of neurons, when Schwann cell serves only to one neuron, one axon. Okay, between these axons we have nodes of Ranvier. Yeah, so nodes of Ranvier. So it's sites where axon is naked, without myelin sheaths. It's necessary for transmission of signal, one-directional transmission of signal. Okay, this is oligodendrocytes, and we have two types of them. So there are intrafascicular oligodendrocytes. Okay. In interfascicular oligodendrocytes, okay, so doesn't matter. So one type of oligodendrocyte. Then we have microglial cells. Microglial cells, they're present between, so in interstitia between neurons, and they're very small, and it seems like they are derived from monocytes. but it seems like it's not confirmed yet. But by function, macroglial cells same as macrophages. They just digest all residues of tissues, might be the cell debris, and due to the injury of cells, so all debris can be digested as microglial cells. And pathology of microglial cells can cause this degenerative diseases like Alzheimer's disease, and also most of degenerative diseases of brain, they're related to microglial cell dysfunction, because they can't clear the debris between nerve tissue. Okay, and last one is ependymal cell, of course. Ependymal cells, they are ependymal cells, they line the ventricles. They're just epithelial cells which are made up of nervous tissue. They're present in ventricles. So 1 minute. 1 minute. So this is you see ependymal cells. They look like epithelial cells, and they're present in ventricles, and also in areas when we need to produce cerebrospinal fluid, CSF. Cerebrospinal fluid is made by ependymal cells. They just filtrate the blood and transport only those components of blood which can produce CSF, cerebrospinal fluid. Okay, and then in peripheral nervous system, we have two types of cells. There are satellite cells and Schwann cells. And usually in most of books, satellite cells are also named as Schwann cells. But a main difference that satellite cells present only around cells in ganglions, so in long bodies of cells. But actually, by derivation, it's the same Schwann cell. But Schwann cells, they're present along the axon, and in some fibers, in some nerve fibers, they form the myelin sheath. So you see, what is a myelin sheath? So myelin sheath is a multiple wrapping of cell membrane around the axon. And actually myelin, it's a structure of cell membrane. Just because of a large thickness of this cell membrane, so it looks like it's fat, yeah. And actually, it's a lipidous compound. But sometimes in some nerves, for example, in parasympathetic and sympathetic nervous system, peripheral nervous system, a lot of fibers are not myelinated. They're not myelinated. It doesn't mean that we have no Schwann cells there. We have Schwann cells, but these Schwann cells are like this. They're present like this. They just wrap around once around the axon, because Schwann cells serve to very important function. They are providing nourishment to axon, because once again, in axon we have a lot of mitochondria, and it also needs some nourishment inside it. And due to this nourishment, so due to providing this nourishment, this axon can act. So that's why Schwann cells present everywhere in myelinated and non-myelinated fibers. So who's calling me right now? Really? Some student late and then calling me to join him in the class? So I don't allow him for model. Actually, I just dismiss his model marks. I don't care. So then, okay, Schwann cells here. So let's talk about connective tissue investments of nervous tissue. It's the last point, and so it's similar classification as we have in muscles. We also have endoneurium, which is present directly around each axon of neuron. It's endoneurium. So it's between different axons. We have endoneurium. It's also reticular fibers. It's reticular fibers which are produced by Schwann cells. We have perineurium. It is which divides the neurons, divides this inside nerve into the fascicles. So you see perineurium. So in analogous to perimysium, yeah. So it divides the entire muscle into fascicles, and here it divides entire nerve into the fascicles. And but blood supplyment goes only between this perineurium. Between this perineurium, which is made by epineurium, which contain surrounds an entire nerve. It is epineurium. It's composed of dense connective tissue. It's very thin, but still it's dense connective tissue, and blood supplyment goes only through the epineurium into these neurons. So because here we have a blood-brain barrier which is made here. Yeah. And also internal nervous system, connective tissue is composed of pia mater, arachnoid membrane, and dura mater. So histologically, there are connective tissues. Pia mater is a loose connective tissue. Arachnoid membrane it's also loose connective tissue, which responsible for production or responsible for blood vessel intake to the nervous system. And dura mater is a dense connective tissue which divides the brain part from bone part of the skull. And we have periosteal dura mater and meningeal dura mater. Meningeal dura mater envelops the brain, periosteal dura mater envelops the bone. Okay, one thing I didn't explain about the blood-brain barrier. Blood-brain barrier is made by astrocytes. I said about it, but I just want to mention this. So this when not everything from blood entered into the cell, into the neuron, it's known as blood-brain barrier. And this blood-brain barrier is very important stuff in administration of drugs. And because neurons, they have this barrier, and that's why some drugs, administration of some drugs is not succeed, and that's why you need to keep it in mind. Okay, that's it. Thank you for attention. So now let's check your attendance. Tayyab Khan, really? So, guys, what's going on in chat? Chat is for attendance. So now I'll record this. I'll record this chat, and everyone who said something, I'll discuss with you personally. Okay. Okay, class is over. Thank you for attention.